Long before Homo sapiens became the only human species left on Earth, other kinds of humans walked beside our ancestors, and some remain frustratingly difficult to identify. A handful of bones, fragments of ancient DNA, and mysterious archaeological traces hint at a population scientists still struggle to place on the human family tree. Were they a forgotten branch of humanity, or something we have yet to fully understand? If you enjoy exploring the deepest mysteries of human origins, like the video, subscribe to the channel and join us as we uncover the lost race of humans scientists still debate. The skull does not look impossible at first. It has a brow ridge, a broad face, heavy bone, and the unmistakable architecture of a human ancestor. Yet the longer researchers study specimens like it, the less comfortably they fit into the family tree. Some seem too modern in one feature and too ancient in another. Some appear in places where no recognized human population should quite exist at that time. Others survive only as fragments, a jaw without a skull, a tooth without a skeleton, a piece of finger bone containing more information in its DNA than generations of fossil hunters once believed possible. Together, these remains have created one of the most unsettling problems in the study of human origins. There may have been entire populations of humans whose place in our evolutionary history is still unresolved. Human evolution was once commonly illustrated as a neat progression, a sequence of increasingly upright and intelligent forms leading toward Homo sapiens. That image was always an oversimplification, but modern discoveries have dismantled it almost completely. The deeper scientists look into the Pleistocene, the geological epoch that stretches from roughly 2.5 million years ago to less than 12,000 years ago, the more crowded the human world becomes. Different human populations overlap across continents. Some split apart, remain isolated for tens of thousands of years, and later meet again. Some exchange genes. Others disappear without leaving enough evidence to reveal who they were. What survives in the record suggests not a ladder but a tangled landscape of branches that repeatedly divide and reconnect. Before we go deeper, take a moment to like the video and subscribe if this is the kind of history and mystery you enjoy. The mystery becomes especially vivid when researchers confront fossils that seem unwilling to accept the labels created for them. A skull found in one region may resemble Homo erectus in its thick cranial bones, while possessing a brain case closer in size to later humans. A jaw may appear surprisingly robust, but come from a period when anatomically modern humans are already present elsewhere. Teeth can be even stranger. Their roots, crowns, and internal structures sometimes reveal relationships that are not obvious from the outside. A single molar can hint that an unknown population once occupied a landscape for thousands of generations. This does not necessarily mean that every unusual fossil represents a separate species. Human bodies vary naturally, and ancient populations were no exception. Age, sex, diet, climate, geographic isolation, and simple individual variation can produce striking differences. Fossils are also incomplete. Pressure from sediment can distort skulls. Chemical processes alter bone. Archaeologists often work with fragments separated from the rest of the skeleton by erosion animals, ancient burials, or sheer chance. Scholars still debate how much anatomical difference is required before an ancient population deserves a new species name. That uncertainty is one reason phrases such as lost race can be misleading if taken literally. In modern scientific language, race is not an appropriate biological category for describing these ancient human groups. The real issue concerns populations, lineages, and possible species within the genus Homo. Yet the older phrase captures something emotionally powerful. The possibility that humans, unlike any living population, once occupied enormous parts of the world, developed their own adaptations, encountered other kinds of humans, and then vanished so completely that even their names are modern inventions. For most of the history of paleoanthropology, bones were nearly everything. Researchers compared skull shape, limb proportions, teeth, stone tools, and geological age. Then ancient DNA transformed the field. Suddenly, a fragment too small to identify visually could reveal an entire evolutionary lineage. The most famous example emerged from Denisova Cave in the Altai Mountains of Siberia. A tiny finger bone, originally unremarkable among thousands of archaeological remains, produced genetic material belonging to a human population different from both modern humans and Neanderthals. These people became known as Denisovans, even though scientists still possess only a sparse collection of physical remains securely associated with them. The discovery changed expectations. If one previously unknown human population could remain hidden until genetics exposed it, others might have disappeared in the same way. Modern genomes soon provided hints that this had indeed happened. Researchers studying patterns of inherited DNA found signals suggesting interbreeding with archaic populations, for which no confidently identified fossil population exists. These are sometimes called ghost populations. The term sounds dramatic, but it describes a technical problem. Genetic evidence indicates that a population contributed ancestry, yet scientists cannot say with certainty what that population looked like, or which archaeological remains belong to it. Africa may contain some of the deepest examples. Because Homo sapiens evolved there, African population history extends enormously far back and cannot be reduced to a single ancestral group moving steadily toward modern humanity. Ancient populations appear to have remained separated across different environments, perhaps for long periods before reconnecting. Forests, deserts, mountains, river systems, and changing climates repeatedly altered where people could move. Genetic models sometimes suggest contributions from deeply divergent lineages, although interpreting these signals is difficult. Population structure within early Homo sapiens can sometimes mimic the genetic signature expected from interbreeding with a different archaic group. No single explanation fully accounts for every pattern. The fossil record makes the picture even less tidy. Across Africa, Europe, and Asia, researchers have discovered remains assigned to names such as Homo heidelbergensis, Homo antecessor, Homo naledi, Homo floresiensis, Homo luzonensis, and several debated classifications surrounding Middle Pleistocene fossils. Some scientists accept particular names as distinct species. Others argue that certain fossils represent regional populations within broader lineages. Taxonomy becomes especially difficult because evolution does not respect the clean boundaries humans prefer to draw on diagrams. A population can gradually change without a clear moment when one species becomes another. Two groups can diverge anatomically, yet remain capable of having children together. Neanderthals and modern humans demonstrate this directly. Denisovans complicate it further. Genetic evidence shows that these populations were separate enough to accumulate major differences, yet not so separate that gene flow became impossible. Their descendants still carry fragments of this ancient contact. That fact changes the meaning of extinction. A human lineage can disappear as a recognizable population while pieces of its ancestry remain alive inside another. The vanished are not always entirely gone, and this is where the mystery begins to deepen. The strange skulls and unexplained genetic signals are not isolated curiosities. They belong to a period when several kinds of humans may have inhabited the planet simultaneously, sometimes separated by thousands of kilometers, sometimes living close enough to meet. To understand the unidentified populations, scientists still debate the familiar image of a world occupied by only one humanity must first be abandoned. The ancient Earth was far more crowded than that. For much of the last few 100,000 years, Earth is not inhabited by a single kind of human. It is a world of overlapping populations separated by mountains, deserts, forests, coastlines, and immense distances, yet never completely sealed from one another. Across Europe, Neanderthals endure repeated cycles of cold and warming. In parts of Asia, Denisovans occupy landscapes that remain only dimly visible through archaeology. In Africa, populations ancestral to Homo sapiens live across environments ranging from grasslands to forests and semi-deserts. Farther east and south, other groups persist whose fossils resist easy classification. The human story during this period is not a march toward one inevitable survivor. It is a shifting mosaic in which several lineages coexist, adapt, migrate, retreat, and occasionally meet. The Pleistocene itself helps create this complexity. Climate repeatedly transforms the continents. Ice sheets expand and contract. Rainfall belts move. Deserts grow, then shrink. Grasslands appear where forest once stood, while coastlines advance and retreat as sea levels rise and fall. For ancient humans, these changes are not background scenery. They determine where animals can be hunted, where fresh water can be found, where plant foods survive, and which migration routes remain open. A region that connects two populations for thousands of years can later become an ecological barrier. When conditions change, again groups that have been separated long enough to develop distinct physical and genetic traits may encounter one another once more. Europe provides the most familiar example. Neanderthals are already established across much of the continent and Western Asia long before Homo sapiens becomes widespread there. They are not primitive caricatures struggling at the edge of survival. Their bodies are powerfully built, well suited to demanding environments, and their archaeological record shows sophisticated behavior. They manufacture carefully prepared stone tools, use fire, hunt larger animals, and occupy caves and open-air sites across enormous territories. Evidence also suggests that at least some Neanderthal groups use pigments, collect unusual objects, and engage in behaviors that may have carried symbolic meaning. Scholars still debate the exact extent of these practices, but the older image of Neanderthals as barely intelligent brutes has become increasingly difficult to defend. East of the traditional Neanderthal range, the picture becomes stranger. Denisovans are genetically distinct, but their geography appears to have been enormous. Their DNA survives in modern populations from parts of Asia, and especially Oceania, indicating that encounters occurred far beyond the Siberian cave where they were first identified genetically. A jaw from the Tibetan plateau has been linked to Denisovans through protein evidence suggesting that some members of this lineage adapted to high-altitude conditions surprisingly early. Certain genetic variants inherited from Denisovan ancestry later helped modern Tibetan populations cope with low oxygen levels. The detail is remarkable. A vanished human population may still influence how living people breathe in one of the harshest inhabited environments on Earth. Yet even Neanderthals and Denisovans may represent only part of the diversity. The fossil record across Asia contains specimens that do not fit neatly into either group. Some skulls preserve a blend of archaic and more derived features. Large teeth from several sites resemble forms associated with Denisovans, but cannot always be assigned confidently. Other fossils have been given new species names, only for researchers to argue that the differences may reflect sex, age, regional variation, or previously unknown members of an established lineage. The evidence remains incomplete because genetic material rarely survives well in warm climates, precisely where some of the most mysterious populations may have lived. Africa is even more complicated. It is the continent where Homo sapiens emerges, but there is no single obvious birthplace marked by a clean sequence of fossils. Early remains with combinations of modern and archaic traits are scattered across distant regions. Some fossils from Morocco, Ethiopia, and southern Africa show that features associated with modern humans appear in a mosaic rather than all at once. A face may look strikingly modern, while the back of the skull retains an older shape. This suggests that the anatomy recognized today as Homo sapiens develops gradually among interconnected populations, rather than appearing suddenly in one isolated community. At the same time, other human lineages may persist alongside these early sapiens populations. Homo naledi, discovered in South Africa, is particularly difficult to fit into simple expectations. It possesses a small brain and several anatomically primitive features, yet dating places its known remains surprisingly late, overlapping in time with early Homo sapiens elsewhere in Africa. Its combination of traits is a reminder that older-looking anatomy does not necessarily mean greater geological age. Evolution does not remove every ancient form the moment a newer one appears. Southeast Asia makes the same point even more dramatically. On Flores, Homo floresiensis survives until relatively late in the Pleistocene. These humans are small-bodied with a distinctive combination of features that has generated intense debate over their ancestry. On Luzon in the Philippines, Homo luzonensis is known from only a small collection of bones and teeth. Yet those remains contain an unusual mixture of characteristics. Both populations live on islands, environments where isolation can produce striking evolutionary changes. Their existence demonstrates that the human world once included forms very different from both Neanderthals and Homo sapiens. Even Homo erectus, one of the longest-lived and most geographically widespread human lineages, appears to survive in parts of Asia far later than earlier models once assumed. On Java, fossils indicate that populations related to Homo erectus persist deep into the late Pleistocene, although the precise dates and relationships remain subjects of continuing research. For a long stretch of prehistory, therefore, Southeast Asia may have contained several different kinds of humans, separated by islands, ecological zones, and changing sea levels. The result is an ancient planet filled with boundaries that are neither permanent nor absolute. Populations remain isolated long enough to become distinctive, but isolation eventually breaks. Rivers change course, grasslands expand, sea levels fall, exposing broad shelves of land. Groups move into territories occupied by others. Some meetings may end in competition, others lead to interbreeding, still others leave no detectable trace at all. This is the deeper reason why the idea of a single lost human race is too simple. There may have been many lost populations, some closely related, some separated for hundreds of thousands of years, and some known only through faint genetic echoes. The closer researchers look, the more the Pleistocene resembles not a stage occupied by one emerging humanity, but an entire world of humanities. And once that crowded world comes into focus, a more difficult problem appears. If so many populations lived at the same time, how can scientists tell where one ends and another begins? The bones provide clues, but some of those clues seem almost deliberately contradictory. Some fossils seem almost designed to frustrate classification. A skull may possess a heavy brow ridge associated with older human forms, but a brain case that falls within the range of later populations. A jaw may lack a chin, suggesting archaic ancestry, yet come from a time when Homo sapiens is already spreading across nearby regions. Teeth can be exceptionally large or strangely shaped, while the rest of the available anatomy points in another direction entirely. These combinations matter because paleoanthropology depends heavily on comparison. When a specimen refuses to resemble any known group closely enough, the neat branches of the human family tree begin to blur. One of the central difficulties is that fossils rarely arrive as complete individuals. The image of an archaeologist uncovering an intact ancient skeleton is far less common than the reality of isolated fragments. A cave may preserve a few teeth, part of a jaw, a piece of skull, or a single limb bone. These remains can be separated from one another by thousands of years of sediment movement. Carnivores may scatter bones. Water may transport fragments. Geological pressure can crush or distort them. Even when researchers recover several pieces from the same site, proving that they belong to one population can be difficult. A species classification may therefore rest on surprisingly limited material. The Middle Pleistocene, roughly the period from several 100,000 years ago into the later stages of human evolution, is especially complicated. Across Africa and Eurasia, fossils appear with mixtures of traits that researchers have historically grouped under names such as Homo heidelbergensis, archaic, Homo sapiens, or other regional classifications. These labels are useful, but they can create the illusion of greater certainty than actually exists. Scholars still debate whether some of these names represent genuine species' broad grades of anatomy or convenient categories covering several distinct populations. The difficulty becomes obvious when skulls from distant regions are placed side by side. Some European fossils appear to show features moving toward the classic Neanderthal form, including changes in the face-brow region and rear of the skull. African specimens of similar age may display different combinations, some eventually associated with the ancestry of Homo sapiens. Asian fossils often complicate both patterns. They may preserve features reminiscent of Homo erectus, while also showing characteristics that seem more derived. No single explanation fully accounts for all of these differences. One particularly useful example comes from the site of Cima de los Huesos in northern Spain. Deep within a cave system, researchers recovered the remains of numerous individuals dating back hundreds of thousands of years. Their anatomy includes several traits associated with later Neanderthals, and genetic evidence places them closer to the Neanderthal lineage than their age might once have suggested. The fossils demonstrate that recognizable Neanderthal features do not appear all at once. They accumulate, gradually producing populations that are neither identical to earlier humans nor fully like the Neanderthals who live much later. This evolutionary mosaic is important because it warns against assuming that every unusual combination marks a completely separate species. Evolution works through populations, and populations can change at different rates in different anatomical regions. Teeth may evolve under one set of pressures while skull shape changes under another. Climate, diet, chewing behavior, body size, genetic drift and isolation can all influence anatomy. A fossil showing an unfamiliar mixture may represent a population in transition rather than an entirely separate branch. Yet some specimens remain difficult to explain even with that caution. China has produced several of the most debated examples. Fossils from sites such as Dalijinyushan, Hualongdong, and other localities exhibit combinations of archaic and more modern-looking traits. Researchers differ over whether these remains belong to regional descendants of Homo erectus early modern humans, Denisovan-related populations, or other groups that have not yet been clearly identified. The problem is made harder by the limited survival of ancient DNA in many warmer environments. Without genetic evidence, anatomical interpretation carries more weight, and different researchers can reasonably emphasize different traits. Teeth have become unexpectedly important in this debate. Dental anatomy often preserves well because enamel is the hardest tissue in the human body. The external shape of a tooth provides information, but modern imaging also allows researchers to examine internal structures, including enamel thickness, dentine shape, and root configuration. Some ancient Asian teeth display patterns that appear similar to Denisovan-associated material. This has raised the possibility that fossils previously impossible to classify may belong to populations related to Denisovans, but resemblance is not proof. Similar traits can evolve independently, and related populations can also retain older characteristics inherited from common ancestors. The evidence remains incomplete. Without secure DNA or proteins, a tooth may indicate a relationship without identifying it conclusively. The same problem appears in Africa. Fossils such as those from Broken Hill, sometimes called Kabwe, preserve a striking mixture of characteristics. The skull is robust with a large brow ridge, yet it also has a large brain case. Its exact place in human evolution has been debated for decades. Similar uncertainty surrounds other African Middle Pleistocene specimens that have been assigned to different species names, depending on the researcher and classification system being used. This uncertainty does not mean scientists are simply guessing. Classification is tested through measurements, comparative anatomy, dating genetics when available, and increasingly through advanced imaging and statistical analysis. But the fossil record is a sample of a sample. Most ancient humans leave no fossil at all. Of those who do, only a tiny fraction are discovered. The result is an evolutionary story reconstructed from scattered pieces of evidence separated by enormous gaps. A lesser-known complication is that researchers must also consider individual variation. Living humans differ substantially in skull shape, tooth size, facial proportions, and body structure, while still belonging to the same species. Ancient populations may have shown equally broad variation, especially when groups remain geographically isolated. A strange skull can therefore look evolutionarily significant, while potentially representing an unusual individual within a known population. Sexual differences add another layer. Male and female skeletons can vary in robustness and proportions. Age changes bones as well. Tooth wear alters the appearance of jaws. Injuries and disease can reshape anatomy. When only one specimen represents a supposed population, these factors become especially important. And yet the pattern does not disappear when all these cautions are considered. Across the Old World, too many unusual fossils remain. Some probably belong to known lineages. Others may represent populations that have not yet been recognized properly. A few could eventually justify entirely new species names. The most interesting possibility is that certain remains may correspond to the ghost populations hinted at by genetics populations whose DNA entered other humans, even though their own physical identity has been lost. The bones therefore create a paradox. They are the most direct evidence that these people existed, yet they can also be the least cooperative evidence for deciding exactly who they were. Every newly discovered fragment has the potential to clarify the family tree, but it can just as easily introduce another branch. And beneath that problem lies an even more fundamental one. Before scientists can decide whether a fossil represents a new human species, they first have to agree on what a species means when different human populations are capable of meeting, mixing, and leaving descendants behind. The word species sounds precise until it is applied to the deep human past. In modern biology, a species is often described as a population capable of reproducing with itself, while remaining reproductively isolated from others. That definition works reasonably well for many living animals, especially when researchers can observe behavior, breeding patterns, and genetics directly. Ancient humans create a much harder problem. The groups under discussion are dead. Their populations are fragmentary. Their boundaries shift through time, and in several important cases, genetic evidence proves that supposedly separate lineages were capable of producing fertile descendants together. The result is one of the most persistent debates in paleoanthropology. How different do two ancient human populations need to be before they should be called separate species? The difficulty is not merely philosophical. Names shape the way evidence is interpreted. If a fossil is assigned to a distinct species, researchers may search for a separate evolutionary origin, geographic range, and behavioral tradition. If the same fossil is considered a regional form of a broader species, the story becomes one of variation within an interconnected population. The bones do not change, but the narrative built around them can change dramatically. Neanderthals are a useful example. They are usually classified as Homo neanderthalensis, a species distinct from Homo sapiens. Anatomically, the differences are substantial. Neanderthals typically possess long, low skulls, prominent brow ridges, large nasal regions, distinctive facial projection, and robust bodies adapted to physically demanding environments. Their skeletons can often be recognized even from partial remains. Yet genetic evidence has shown that Neanderthals and Homo sapiens interbred repeatedly. Many living people outside Africa carry a small percentage of Neanderthal ancestry, and some African populations also possess Neanderthal-derived DNA through later migrations back into the continent. If separate species can interbreed, the simple biological definition begins to strain. This is not unique to humans. Closely related animal species can sometimes hybridize. Wolves, coyotes, and domestic dogs provide familiar examples of gene exchange across taxonomic boundaries. Bears can also hybridize under certain conditions. Species separation is therefore not always an absolute wall. It can be a gradual process, with populations becoming increasingly different while limited gene flow remains possible. For ancient humans, this gradual separation may have been common. Neanderthals, Denisovans, and Homo sapiens diverged from related ancestral populations over hundreds of thousands of years, but they did not become completely reproductively isolated. Denisovans themselves appear to have exchanged genes with Neanderthals and with modern human populations. Genetic studies even suggest that some Denisovan groups may have been deeply separated from one another. The label Denisovan may therefore conceal more diversity than a single name implies. This is where the species problem becomes especially important for the mysterious populations scientists still debate. A strange skull may differ enough from known specimens to look like a new species, but if ancient human populations were structured as interconnected regional groups, then anatomical differences may reflect long isolation without complete separation. Two populations can look quite different and still exchange genes when geographic barriers disappear. Paleoanthropologists therefore use several different approaches. Some emphasize morphology, the measurable anatomy of bones and teeth. If a group consistently displays a distinctive combination of traits not found in others, that pattern may support recognition as a separate species. Others emphasize evolutionary ancestry, asking whether a population represents its own branch with a distinct historical trajectory. Geneticists can examine divergence times and gene flow where ancient DNA survives. Archaeologists add information about geography, behavior tools, and chronology. None of these methods alone provides a perfect answer. The problem becomes clearer with Homo heidelbergensis. For decades, the name has been used for a wide range of Middle Pleistocene fossils from Africa and Europe and sometimes Asia. In some models, Homo heidelbergensis is treated as a widespread ancestor of Neanderthals and modern humans. In others, European fossils assigned to this name are more closely tied to Neanderthal ancestry, while African specimens deserve a different classification. Some researchers avoid the term for certain fossils altogether because it can combine anatomically and evolutionarily different populations under one convenient label. The debate is not simply about terminology. If the fossils grouped under Homo heidelbergensis actually represent several distinct lineages, then human diversity during the Middle Pleistocene is greater than once assumed. If they are regional populations within a broad connected species, then the story instead emphasizes gene flow across a vast geographic range. Scholars still debate which model best matches the surviving evidence. Similar disputes surround other names. Homo rhodesiensis has been proposed for certain African Middle Pleistocene fossils, though its usage remains controversial. More recently, some researchers have suggested Homo bodoensis as a way to reorganize confusing classifications, but the proposal has not achieved universal acceptance. In East Asia, names such as Homo longi have entered the discussion following the study of the Harbin skull. Each new name attempts to describe real anatomical patterns, but each also risks creating additional branches where the evolutionary landscape may have been more continuous. There is an irony in this. Taxonomy requires boundaries, but evolution often produces gradients. Imagine ancient populations distributed across thousands of kilometers. Groups living at opposite ends of that range may differ noticeably in anatomy. Neighboring groups, however, may remain similar and occasionally exchange mates. Over many generations, traits can change gradually across geography. Where should one species end and another begin? The fossil record captures only scattered points along that continuum, making gradual variation appear as abrupt gaps. Time creates the same problem. A lineage may change slowly across hundreds of thousands of years. Earlier and later populations can look different enough that scientists give them separate names, even though no generation ever gives birth to an offspring belonging suddenly to a new species. Species are useful scientific categories, but nature does not produce labels. A lesser-known complication comes from the concept of introgression. When two diverged populations interbreed, genes can cross from one lineage into another and remain there for thousands of generations. This process has been documented among Neanderthals, Denisovans, and Homo sapiens. It means that even populations with long independent histories can share important genetic material. Some inherited variants may disappear while others spread because they provide useful adaptations. This helps explain why anatomy, genetics, and species names do not always align perfectly. A population can inherit genes from another without becoming anatomically identical to it. Conversely, two groups can develop similar features under similar environmental pressures despite limited genetic relationship. The fossil record therefore records both ancestry and adaptation, and separating the two can be extremely difficult. The question of what constitutes a species becomes even more complicated when only one or two fossils are available. Homo luzonensis is known from a limited number of remains. Homo floresiensis has a much better record, but its ancestry is still debated. The Denisovans were genetically recognized before scientists had anything close to a complete skull securely associated with them. In each case, the evidence forces researchers to classify populations that are only partially visible. This uncertainty is not a weakness of the science. It is a consequence of studying a world in which almost all biological evidence has disappeared. Researchers build hypotheses from what survives, compare competing explanations, and revise classifications when new discoveries demand it. But the species debate leaves one possibility increasingly difficult to ignore. If human evolution operated through long-separated populations that sometimes reconnected, then some ancient groups may never fit comfortably into a single named box. They may have been distinctive enough to leave genetic or anatomical traces, yet connected enough to exchange ancestry with neighboring humans. And genetics is beginning to reveal exactly such populations, not through skulls or skeletons, but through fragments hidden inside people alive today. Some of the most important ancient humans ever discovered have never been identified from a complete skeleton. They have no reconstructed face, no securely associated burial, and in some cases no fossil that scientists can point to and say with confidence this is one of them. their existence is inferred from something far less visible, but in many ways more revealing fragments of DNA preserved inside other populations. These are the so-called ghost populations, ancient human groups that contributed ancestry to later people, but remain archaeologically elusive. They represent one of the strangest consequences of the genetic revolution. The human past contains individuals whose descendants survive even though the people themselves have almost disappeared from the physical record. The discovery of Denisovans demonstrated how dramatically genetics could expose such hidden histories. Before DNA was extracted from material in Denisova Cave, there was no recognized human group called Denisovans. The small finger bone that transformed the field did not look like the remains of an entirely unknown branch of humanity. Its importance was molecular. Genetic sequencing showed that the individual belonged to a lineage related to but clearly distinct from Neanderthals. A population had effectively been discovered through its genome before researchers knew what its members looked like. Once that possibility became real, scientists began searching modern and ancient genomes for similar signals. The basic idea is deceptively simple. When two populations separate, their DNA gradually accumulates differences. If those populations later meet and interbreed, fragments of one lineage enter the genome of the other. Over generations, those fragments become shorter as chromosomes recombine, but some can survive for tens of thousands of years. By comparing patterns across living populations and ancient genomes, researchers can identify pieces that seem unusually divergent and may have come from archaic humans. In practice, the process is extraordinarily complicated. Human populations have repeatedly split, migrated, mixed, shrunk, expanded, and reconnected. A genetic pattern that appears to come from an unknown archaic population might instead result from deep population structure within Homo sapiens. Statistical models can produce different interpretations depending on assumptions about population size, divergence, dates, and migration. Scholars still debate several proposed cases of ghost ancestry, especially in Africa, where environmental conditions have made the recovery of very ancient DNA extremely difficult. Even so, some patterns are difficult to explain without deep ancestral complexity. Studies of African genomes have identified genetic segments that appear more divergent than expected under simple models of modern human history. One possibility is that ancestors of living Africans interbred with archaic human populations that had separated from the lineage leading toward Homo sapiens hundreds of thousands of years earlier. Another possibility is that early Homo sapiens populations themselves were highly structured, remaining partly isolated for long periods before later merging. Both scenarios create similar genetic signatures, and the evidence remains incomplete. This distinction matters. Calling an unidentified contributor an archaic species can make the story sound clearer than it really is. The unknown population may have belonged to a deeply separated human lineage, or it may have been part of a broad network of early sapiens populations that developed substantial differences while remaining connected through occasional gene flow. Genetic data can reveal that two ancestral groups were distant. It cannot always tell researchers what names those groups would deserve if their bones were available. West and Central Africa have drawn particular attention because some genomic studies suggest deeply divergent ancestry there. Researchers have proposed that unknown populations may have contributed genetic material to the ancestors of certain present-day groups. Estimates of when those lineages diverged vary considerably across studies and models. What survives in the record suggests a complicated history rather than a single dramatic encounter with one mysterious species. Ancient Africa was large, environmentally diverse, and inhabited by human populations for immense periods of time. Long-term regional separation followed by repeated contact is therefore entirely plausible. Similar mysteries exist outside Africa. Denisovan ancestry itself is not uniform. Modern populations in Oceania, East Asia, and parts of Southeast Asia carry different patterns of Denisovan-related DNA. These differences suggest that Homo sapiens may have encountered genetically distinct Denisovan populations on more than one occasion. In other words, the Denisovans may not have been one homogeneous group. Their lineage could have contained regional populations separated from one another for substantial spans of time. That possibility radically expands the mystery. The small number of confirmed Denisovan remains comes from only a handful of locations, while their genetic legacy stretches across enormous distances. Somewhere between Siberia, the Tibetan Plateau, mainland Asia, and the islands farther south, Denisovan-related populations must have lived in landscapes where their bones have not yet been clearly recognized. Fossils may already exist in museum collections under different classifications. Others may still lie undiscovered in caves or sediment deposits. Some may never have fossilized at all. A particularly surprising detail is that researchers can sometimes identify archaic contributions even without possessing a reference genome from the contributing population. Statistical methods can search for DNA segments that do not fit expected patterns among known populations. These segments are sometimes described as unusually deep branches in the genome. Their existence does not automatically prove a separate species, but they can reveal ancestral relationships that fossils alone might never expose. This is one reason the concept of extinction becomes increasingly complicated. Imagine a small archaic population living near a larger expanding population of Homo sapiens. Over generations, interbreeding occurs. The smaller group gradually declines as a distinct community. Its language, appearance, and social identity vanish. Eventually, no separate population remains. From an archaeological perspective, it may look extinct. Genetically, however, portions of its ancestry continue inside the expanding group. There does not need to be a final moment when the last isolated member dies. A population can disappear through absorption. That process may have occurred repeatedly. Neanderthals clearly contributed ancestry to Homo sapiens. Denisovans did the same. Unknown populations may also have been absorbed into larger groups, leaving behind only diluted genetic traces. Some of these fragments may later become common because they are useful. Others persist simply because chance allows them to survive. Yet genetic survival does not guarantee that scientists can reconstruct the vanished population. DNA may reveal that an unknown lineage existed, and roughly when it separated from others, but it cannot automatically show its face, body proportions, technology, language, or culture. A ghost population remains human history seen through a narrow keyhole. The fossils discussed earlier may eventually provide the missing connection. An unusual skull from Africa or Asia might one day yield proteins or DNA linking it to one of these mysterious genetic signals. But until that happens, the relationship between unnamed genomes and unnamed bones remains uncertain. And nowhere is that uncertainty more profound than in Africa, where the deepest history of Homo sapiens appears to have unfolded not within a single isolated birthplace, but across a continent of populations whose boundaries repeatedly formed, shifted, and disappeared. Africa does not preserve the beginning of Homo sapiens as a single clean event. There is no one valley, one population, or one perfectly timed fossil that can be pointed to as the unquestioned birthplace of modern humanity. Instead, the evidence is scattered across a continent vast enough to contain deserts, tropical forests, savannas, mountains, wetlands, and long coastlines, all of them shifting dramatically over hundreds of thousands of years. Fossils with increasingly modern traits appear in different regions at different times. genetic evidence points toward deep population structure, archaeology reveals technologies that spread unevenly. Taken together, the pattern suggests that early Homo sapiens may have emerged through a long process of interaction among populations that were sometimes connected and sometimes separated for enormous spans of time. One of the clearest examples comes from Jebel Irhoud in Morocco, where fossils dated to more than 300,000 years ago possess a striking combination of features. Their faces look surprisingly modern in several respects, yet the shape of the brain case remains more elongated than in most humans living today. This combination matters because it shows that the anatomy associated with Homo sapiens does not appear as a complete package. Different parts of the body change at different rates. A recognizable modern face can exist while other aspects of the skull retain older characteristics. Far to the east, fossils from Ethiopia add another layer. Remains from Omo Kibish and Herto document early Homo sapiens in eastern Africa, but they do not create a simple sequence leading neatly from one form to the next. Instead, they show variation across time and geography. Other African fossils remain even harder to classify. Some possess large brow ridges, robust skulls, or unusual combinations of archaic and modern traits. Scholars still debate whether these individuals belong within early Homo sapiens, represent neighboring populations, or preserve branches that eventually disappear. The landscape helps explain why such diversity could develop. During wetter climatic phases, regions that are now dry may support lakes, rivers, grasslands, and animal populations. The Sahara today, one of the greatest barriers on Earth, periodically becomes far greener. At other times, aridity expands, dividing human communities. Forest belts grow or retreat. River systems change. Populations that once exchanged people, tools, and genes can become isolated as ecological corridors close. Isolation does not need to be complete to have evolutionary consequences. Even partial separation lasting thousands of generations can allow genetic differences to accumulate. Small populations are especially vulnerable to genetic drift, in which traits become common or disappear largely by chance. Local diets, diseases, temperature, altitude, and available resources can push populations in different directions. When environmental conditions later reconnect those groups, the result is not necessarily replacement. They can meet exchange genes and combine lineages that have been developing separately. This model is sometimes described as a structured or Pan-African origin for Homo sapiens. It does not mean modern humans evolved everywhere in Africa equally, or that every ancient African population contributed the same amount of ancestry. Rather, it emphasizes that the continent may have contained several connected population centers instead of one permanently isolated cradle. The exact geography and timing remain under investigation, but the concept fits increasingly well with a record that refuses to collapse into a single straight line. The surprising implication is that some of the unusual fossils once treated as evolutionary dead ends may instead represent populations participating in a much broader network. Their descendants might contribute only small amounts of ancestry to later people, while their distinctive anatomy disappears. Other populations may truly vanish without descendants. Still others could remain isolated long enough to look almost like separate species before reconnecting with populations closer to early Homo sapiens. Genetic data from living Africans has become central to investigating these possibilities. Because as African populations contain extraordinarily deep human genetic diversity, researchers can use modern genomes to reconstruct parts of the past that ancient DNA has not yet preserved directly. Several studies have identified patterns consistent with long-term separation among ancestral populations. Some models also detect signals that could result from introgression by a deeply diverged archaic group. Here the uncertainty becomes crucial. A deeply divergent genetic signal does not automatically prove that an unknown species of human survived in Africa until recently enough to interbreed with Homo sapiens. Similar signatures can be generated by population structure within the ancestors of modern humans. Imagine two populations separating for a very long time, accumulating genetic differences and then merging. Their reunion can resemble a mixture between separate species, even though both populations belong to a broader evolving Homo sapiens lineage. No single explanation fully accounts for every genetic pattern, and different analytical methods sometimes produce different estimates. This is one reason the phrase ghost population must be used carefully. The ghost is statistically inferred, but its biological identity can remain unknown. It may represent a population so distinct that many scientists would classify it outside Homo sapiens. It may represent an ancient branch within early sapiens diversity. It may even represent several populations whose separate histories become blurred into one signal when reconstructed through modern genomes. Africa's fossil record contains candidates that make these possibilities difficult to dismiss. Homo naledi survives surprisingly late in southern Africa, though there is currently no strong evidence showing that it contributed ancestry to modern humans. Other robust Middle Pleistocene fossils represent populations whose relationships remain debated. Because tropical heat and humidity destroy DNA rapidly, many of these fossils may never yield genomes. Researchers therefore turn increasingly to proteins' dental structures, high-resolution imaging, and improved dating methods. The archaeological record adds another warning against oversimplification. Technologies and behaviors associated with the Middle Stone Age appear across Africa in complicated regional patterns. Stone points prepared core technologies, pigments engraved objects, shell beads, and long-distance material transport do not all emerge in one place at one moment. Some disappear and later reappear. Others develop independently or move between regions. What survives in the record suggests human communities repeatedly innovating under different conditions rather than a single cultural revolution spreading outward from one center. A lesser-known feature of this story is just how small some ancient populations may have been. Human groups can occupy enormous geographic ranges while still existing at relatively low densities. Local extinctions may therefore happen frequently. A drought lasting generations could empty an entire region. A population might survive elsewhere, return later, and encounter descendants of people from whom it separated thousands of years before. Over evolutionary time, Africa could repeatedly lose and rebuild connections among human communities. This helps explain why human ancestry can be both deeply divided and surprisingly mixed. The same continent that creates barriers also repeatedly removes them. By the time Homo sapiens begins expanding more successfully beyond Africa, the population carrying that name may already contain ancestry from many older African lineages. Some contributions become widespread, others remain regional, some disappear through drift. The final result is not one pristine ancestral population, but a species assembled across deep time through separation and reunion, yet Africa is only part of the unresolved picture. Far to the east, another group of fossils presents an even more visible challenge. Their skulls are large, their features are puzzlingly mixed, and some may belong to the Denisovans lineage whose face scientists have spent years trying to find. Across East Asia, the human fossil record becomes unusually difficult to read. The problem is not a lack of evidence. In fact, the region has produced an impressive collection of skulls, jaws, teeth, and partial skeletons spanning hundreds of thousands of years. The difficulty is that many of these fossils refuse to fit comfortably into the categories built from discoveries in Europe and Africa. Some preserve features associated with Homo erectus. Others look unexpectedly close to later humans. A few combine both patterns in the same individual. As more fossils are studied with modern imaging, improved dating, and comparative anatomy, one possibility becomes increasingly difficult to ignore. East Asia may have preserved several ancient human populations whose identities are still unresolved. For much of the 20th century, many Asian fossils were interpreted through the long history of Homo erectus. This made sense. Homo erectus, or populations closely related to it, had occupied parts of Asia for an extraordinary span of time. Fossils from Java and northern China demonstrated that early humans reached the region more than 1 million years ago. At Zhoukoudian near Beijing, the famous remains historically known as Peking Man helped establish the importance of East Asia in the human story. Their long low skulls, heavy brow ridges, and thick cranial bones became part of the classic image of Asian Homo erectus, but later fossils complicated that picture. Some specimens dating to the Middle and Late Pleistocene retained robust archaic features while possessing larger brain cases and facial proportions that looked different from earlier Homo erectus. These humans seem neither fully like the older populations nor comfortably identical to Homo sapiens. Researchers sometimes refer to them descriptively as archaic or Middle Pleistocene Homo, but those broad labels conceal considerable variation. The Dali skull from central China is one of the best-known examples. Its brain case is large compared with classic Homo erectus, while its face combines robust and more derived characteristics. Various researchers have interpreted it differently over the decades. Some emphasize continuity with earlier East Asian populations. Others see similarities with early Homo sapiens or other Middle Pleistocene groups. The fossil is important precisely because it does not produce an easy answer. Jinishan in northeastern China presents another complicated combination. The partial skeleton includes a relatively large cranial capacity and anatomical features that have been interpreted in different ways. At Hualongdong, fossils dated to roughly 300,000 years ago add further evidence of human populations displaying a mosaic of traits. The Hualongdong remains include facial characteristics that some researchers consider unexpectedly derived for their age, while other parts of the anatomy retain older patterns. Scholars still debate how these fossils relate to Homo sapiens denisovans and other Asian lineages. Then there is the skull from Harbin in northeastern China, one of the most visually striking fossils to enter the debate. It is massive with a long, low cranial form and substantial brow ridges, yet it also possesses a large brain case and a broad face with a combination of features that prompted researchers to propose the species name Homo longi. Popularly, the fossil became associated with the label Dragon Man. Its exact evolutionary position remains disputed, and later comparisons have raised the possibility that it could be closely related to denisovans. That possibility is tantalizing because denisovans remain anatomically elusive. Genetics confirms that they were a major human lineage, but the number of fossils securely linked to them remains tiny compared with the record for Neanderthals. If some of the mysterious Middle Pleistocene skulls from China belong to Denisovan-related populations, then fossils that have sat outside a clear classification may suddenly acquire an evolutionary identity. Teeth strengthen this suspicion. Denisovan remains from Siberia include unusually large molars, and dental evidence from elsewhere in Asia has drawn comparisons with them. A mandible from the Tibetan plateau known as the Xiahi mandible has been linked to Denisovans through ancient protein analysis. Its morphology shows that Denisovan-related humans were capable of living at high altitude long before Homo sapiens became established there. This dramatically expands the known ecological range of the lineage. The Tibetan Plateau is not an obvious place for an archaic human population to thrive. Oxygen levels at low temperatures can be severe, and resources are seasonally limited. Yet Denisovan-related ancestry later contributed a genetic variant associated with high-altitude adaptation to the ancestors of modern Tibetan populations. The implication is extraordinary. A vanished population may have spent enough time in high-altitude environments for natural selection to shape useful adaptations, and later humans inherited part of that biological solution through interbreeding. Farther south, the mystery deepens because climatic conditions make ancient DNA much harder to preserve. Fossils recovered from caves in southern China and Southeast Asia therefore often have to be studied without genetic confirmation. Some teeth and skull fragments may belong to Denisovan-related humans. Others may represent early Homo sapiens or populations descended from older Asian lineages. Without molecular evidence, researchers must rely on anatomy-geological context dating and increasingly the study of ancient proteins. A lesser-known example of how complicated this can become comes from unusual dental remains. Tooth crowns can appear broadly similar while their internal structures differ. High-resolution scans allow researchers to examine the boundary between enamel and dentine, the number and shape of roots and patterns formed during tooth development. These traits can preserve evolutionary signals invisible to the naked eye. In some Asian fossils, internal dental anatomy has suggested relationships not obvious from the external shape alone, yet teeth also illustrate the danger of overconfidence. Large molars are not uniquely Denisovan. Robust jaws are not exclusive to a single lineage. Similar diets and developmental histories can produce overlapping traits. A fossil may resemble one population in its teeth and another in its skull. The evidence remains incomplete, and classification frequently changes when new material becomes available. There is also a geographic problem. East Asia is enormous. Treating its ancient inhabitants as one continuous regional population risks hiding local histories. Northern China, the Tibetan Plateau, southern China, Southeast Asia, and the islands farther south contain dramatically different environments. Populations separated across these regions may have experienced long periods of isolation. The humans living in one ecological zone might be related to those elsewhere without being identical to them. Genetic evidence from living people supports this broader complexity. Different modern populations carry different patterns of Denisovan-related ancestry, suggesting more than one episode of contact and possibly encounters with more than one Denisovan population. The lineage identified at Denisova Cave may therefore be only one regional representative of a much wider family of archaic Asian humans. This raises an unsettling possibility. There may never have been one simple Denisovan appearance. Just as Homo sapiens populations today display physical variation across geography, Denisovan-related groups spread across Asia may also have differed substantially from one another. A northern population could look different from one adapted to high altitude, and both could differ again from groups farther south. If that is correct, researchers searching for a single definitive Denisovan skull may be asking the wrong question. The unknown faces of ancient Asia may belong not to one lost population, but to a network of related groups, each carrying part of the ancestry modern genetics now calls Denisovan. And the most frustrating part is that scientists already know this lineage was real. They can trace its DNA across thousands of kilometers. They can identify pieces of its biological legacy in living people. Yet after years of study, the question of what a Denisovan truly looked like remains only partially answered. For a population that has transformed the study of human evolution, Denisovans remain almost faceless. Neanderthals are represented by skulls, skeletons, tools, injuries, burial contexts, and enough anatomical evidence for artists and scientists to reconstruct their appearance with reasonable confidence. Denisovans are different. Their existence is certain, their genetic importance is enormous, and their geographic reach appears surprisingly broad, yet their physical form is still reconstructed from a sparse collection of remains. This imbalance between genetic certainty and anatomical absence is what makes them one of the strangest human populations ever identified. The story begins in Denisova Cave in the Altai Mountains, where archaeological layers preserve evidence from different human groups over long periods of time. The cave is not remarkable because it contains one dramatic skeleton. Its importance comes from fragments. A finger bone, several teeth pieces of bone that are almost impossible to identify visually, and sediment containing ancient genetic material have gradually revealed that Neanderthals, Denisovans, and modern humans all occupied or passed through the broader region at different times. The cave becomes less like the home of one population and more like a crossroads used repeatedly by changing groups. The small finger bone that first exposed the Denisovan lineage belonged to a young individual. When researchers extracted mitochondrial DNA, the sequence was unexpectedly different from both modern humans and known Neanderthals. Nuclear DNA later confirmed that the individual belonged to a sister lineage of Neanderthals. The discovery was extraordinary because nothing about the fragment itself announced that it came from a previously unknown human population. Genetics did what anatomy alone could not. Later discoveries made the picture more complex. Large molars from the cave were associated with Denisovans, and their size attracted attention because they looked unusual compared with many recent humans. But teeth alone cannot provide a face. A fragment of skull might reveal part of cranial shape, yet the available material remains too incomplete to define a typical Denisovan head. Even the idea of a typical appearance may be misleading if Denisovan populations were distributed across vast regions and had been separated from one another for long periods. One of the most remarkable findings from Denisova Cave does not belong cleanly to any single lineage. A small bone fragment was genetically identified as belonging to a girl whose mother was Neanderthal and whose father was Denisovan. The individual, sometimes informally known as Denisova 11, provides direct evidence that these groups did not merely share distant ancestors. They met and had children. Her Denisovan father also carried traces of Neanderthal ancestry, from an earlier mixing event indicating that interbreeding occurred more than once. This single fragment changes how the cave can be imagined. The Altai is not simply a boundary where separate human species avoid each other. It is a contact zone. Populations move through the same landscape at different times, sometimes replacing one another, sometimes overlapping and occasionally mixing. What survives in the record suggests a level of interaction that would have been difficult to predict from fossil anatomy alone. The mystery expands dramatically beyond Siberia. Genetic evidence in living populations shows that Denisovan ancestry is especially significant in parts of Oceania and occurs at lower levels across various Asian populations. That distribution cannot be explained by a lineage confined to the Altai Mountains. Denisovan-related humans must have lived across much wider areas, perhaps including parts of East Asia and Southeast Asia. Homo sapiens encountered them somewhere along migration routes through those regions, and these meetings left descendants. The Jahi mandible from the Tibetan plateau provides one of the strongest physical clues. Found in Baishia Karst cave, it could not initially be linked through ancient DNA, because preservation conditions were unfavorable. Instead, researchers analyzed ancient proteins preserved in a tooth and identified similarities consistent with Denisovan affiliation. The jaw is robust, and its presence at high altitude indicates that Denisovan-related humans occupied the plateau surprisingly early. This matters for another reason. Some living Tibetan populations carry a version of a gene associated with adaptation to low-oxygen environments that appears to have been inherited from Denisovan-related ancestors. In evolutionary terms, Homo sapiens may have entered a difficult environment already containing humans who had spent far longer adapting to it. Interbreeding then allowed useful genetic material to move between populations. Evolutionary adaptation in this case may have been partly acquired rather than built from the beginning, but the Tibetan Denisovans may not have looked exactly like the Siberian ones. The distances involved are enormous, and genetic studies suggest that Denisovan ancestry in modern humans may derive from several distinct episodes of interbreeding. Some analyses support the existence of deeply separated Denisovan-related populations. If so, the term Denisovan is functioning as an umbrella for a lineage containing internal diversity, much as the term Homo sapiens includes populations adapted to very different environments today. That possibility makes reconstruction extremely difficult. A face inferred from Siberian genetic material might not represent populations farther south. A jaw from Tibet could belong to one branch of a larger Denisovan network. A mysterious skull in China might be related to another. The evidence remains incomplete, and scholars still debate how these fossils should be connected. Researchers have attempted to infer Denisovan anatomy indirectly from patterns of gene regulation and other molecular clues, but such reconstructions carry substantial uncertainty. Genetics can suggest differences in skeletal development, yet it cannot simply generate a reliable portrait of a vanished population. Physical appearance results from complicated interactions among many genes, development, environment, nutrition, age, and sex. A molecular reconstruction can provide hypotheses, not a photograph. This is why fossil evidence remains essential. One well-preserved Denisovan skull from a securely dated context could transform the field. It could allow researchers to compare debated Asian fossils directly, test proposed relationships, and perhaps resolve whether specimens such as Harbin or others belong near the Denisovan branch. Until such evidence appears, scientists must work backward from scattered pieces. A lesser-known but powerful technique now helps with that search: proteomics. This study of ancient proteins can sometimes identify evolutionary relationships in fossils too old or too poorly preserved for usable DNA. Proteins generally survive longer than DNA under certain conditions. The method cannot provide the same wealth of information as a complete genome, but it may connect isolated bones and teeth to known lineages. The Xiahi mandible shows how valuable this can be. Sediment DNA adds another dimension. Researchers can sometimes recover ancient human genetic material directly from cave sediments, even when no recognizable human bone is present. This means a population can be detected at a site through traces left in the environment. The archaeological landscape becomes populated by genetic shadows. Denisovans therefore force scientists to confront an unusual situation. A human lineage can be mapped genetically across continents before its anatomy is understood. It can contribute adaptive genes to living populations while leaving almost no recognizable skeletal record. It can be known through descendants more clearly than through its own remains. Somewhere among the fossils already excavated from Asia may be the face researchers are searching for. It may be stored under another classification waiting for protein's DNA or better comparative methods to reveal its identity. And one candidate has already attracted extraordinary attention: a massive skull from northeastern China, whose combination of features has reopened the debate over how many kinds of humans once lived across Asia. The Harbin skull appears almost too complete for a fossil at the centre of such uncertainty. Its broad face, enormous brow region, long low vault and large internal brain volume immediately distinguish it from a typical modern human skull. Yet it does not resemble classic Neanderthals closely enough to fit comfortably within that lineage, and it does not match the traditional image of Homo erectus either. When researchers described it in detail, they proposed that it represented a separate species Homo longi, a name derived from the region associated with the fossil, and later popularized through the dramatic nickname Dragon Man. Almost immediately, however, another possibility began to attract attention. Perhaps this was not an entirely new branch at all. Perhaps the skull belongs somewhere near the elusive Denisovans. The fossil itself has an unusual history. According to the account associated with its study, the skull was reportedly discovered decades earlier during construction activity near Harbin in northeastern China and remained hidden for many years before reaching researchers. That history creates complications because the skull was not excavated from a carefully documented archaeological layer. Scientists can study its anatomy and use geochemical and contextual methods to estimate aspects of its history. But the absence of a secure original excavation context limits certainty about its exact age and association with surrounding sediments or artifacts. Even so, the specimen is exceptional. The skull is large and robust with a broad facial structure and substantial brow ridges. Its cranial capacity falls within a range comparable to modern humans and other large brain members of Homo. At the same time, several features retain an archaic appearance. This mixture immediately places Harbin among the mosaic fossils that complicate Middle Pleistocene human evolution. The researchers who formally described Homo longi argued that the fossil represented a distinct lineage close to Homo sapiens. Their analyses suggested that Harbin and certain other Chinese fossils might form a branch separate from Neanderthals and different from earlier Homo erectus. Under that interpretation, human evolution in Asia includes another major lineage whose importance had previously been underestimated. But phylogenetic analyses depend heavily on which anatomical traits are measured, how those traits are coded, which fossils are included, and what assumptions are built into the model. Another team can examine overlapping evidence and produce a different family tree. Scholars still debate the exact position of Harbin, and not everyone accepts Homo longi as a valid separate species. The Denisovan possibility is especially compelling because of geography and anatomy. Confirmed or strongly supported Denisovan-related remains now occur in Siberia and on the Tibetan Plateau, while genetic evidence implies a much wider distribution across Asia. It would therefore be unsurprising if Denisovan populations also occupied regions of northern China. Harbin lies within a broad geographic zone where such contact is plausible. The problem is that there is no complete Denisovan skull available for direct comparison. Scientists know Denisovans mainly from genetic material and limited skeletal remains. The Xiahi mandible offers valuable anatomical information, but it lacks the upper face and cranial vault needed to establish a complete Denisovan skull pattern. Teeth from Denisova Cave add another set of clues, but they cannot determine whether Harbin's facial form is characteristic of the lineage. Thus, identifying Harbin as Denisovan requires an indirect argument known Denisovan geography, molecular relationship similarities among certain Asian fossils, and the expectation that a lineage this widespread must have left larger skeletal remains somewhere. Some researchers have suggested that Harbin could be closely related to Denisovans, perhaps even representing one of their regional populations. Others prefer to keep Homo longi separate until molecular evidence establishes a connection. The evidence remains incomplete, and a successful extraction of ancient DNA or sufficiently informative proteins from the skull could dramatically change the debate. This uncertainty extends beyond Harbin itself. Several Chinese fossils have been compared with it, including specimens from Dali Jinyashan and other sites. If these remains form a coherent lineage, scientists must determine whether that lineage is Homo longi, Denisovan, a regional form of another group, or something more complex. If they do not form one lineage, then East Asia may contain several different human populations that happen to share some broad anatomical characteristics. This situation exposes a recurring weakness in the search for lost human species. One striking fossil can appear to solve a mystery while actually creating several new ones. Species names are especially vulnerable to this problem. Naming a fossil can be useful because it gives researchers a framework for comparison. But a name can also imply a biological boundary stronger than the evidence supports. Homo longi may eventually prove to be a meaningful evolutionary group. Alternatively, it may become absorbed into a broader Denisovan classification. Scientific names are hypotheses about relationships as much as labels attached to bones. A surprising detail is that the Harbin skull's large brain does not make its owner more modern in any simple evolutionary sense. Brain size varies significantly among ancient human groups, and Neanderthals often possessed cranial capacities comparable to or larger than the modern human average. Evolutionary relationships depend on structure development ancestry and numerous anatomical characteristics, not on a straightforward progression toward larger brains. The face is equally deceptive. Some features that appear modern can emerge independently or persist from older ancestral populations. A broad flat face, reduced projection, or particular cheekbone arrangement may suggest relationships, but each trait must be interpreted within the whole anatomical pattern. Mosaic evolution repeatedly produces combinations that make simple visual classification unreliable. Dating adds another layer. Human populations in East Asia survive across immense spans of time, and a skull's evolutionary meaning depends partly on exactly when its owner lived. A morphology that seems surprisingly advanced at one age may be less unusual at another. Because Harbin lacks a secure, excavated context, estimates of its age require caution, even though evidence places it within the Middle Pleistocene or later part of that broad interval. What survives in the record suggests a human world in East Asia that was anything but empty or static. Populations descended from early dispersals had occupied the continent for hundreds of thousands of years. New groups arrived, others became isolated. Some perhaps mixed with Denisovan's Neanderthal-related populations or early Homo sapiens. By the time modern humans expanded more widely through the region, they may have encountered a landscape already inhabited by several forms of humanity. This possibility changes the significance of Dragon Man. The skull matters not because it proves a sensational lost race, but because it demonstrates how little certainty exists about the humans who occupied Asia during one of the most important periods of our evolution. Harbin could represent a distinct species. It could be Denisovan. It could belong to a broader regional lineage that scientists have not yet defined properly. None of these possibilities has been settled beyond dispute, and Harbin is still a large, familiar-looking human skull. Farther south on isolated islands, evolution produced something even harder to anticipate: small-bodied humans, whose anatomy seemed at first so unexpected that some researchers questioned whether such populations could really have existed at all. On isolated islands, human evolution produces some of its strangest experiments. The populations that reach these places do not necessarily remain miniature versions of their ancestors. Isolation changes the rules. Food may be limited, predators may disappear, body size can shift, and small founding populations can carry unusual genetic traits that become common over generations. In Southeast Asia, this process appears to have created human forms so unexpected that their discovery forced scientists to reconsider how diverse the genus Homo had become. Homo floresiensis and Homo luzonensis are not merely curiosities. They demonstrate that ancient humanity could survive in forms very different from both Homo sapiens and Neanderthals, and they raise a difficult question about how many other isolated populations vanished before anyone knew they existed. The most famous case comes from the Indonesian island of Flores. In Liangbua Cave, archaeologists uncovered the partial skeleton of a remarkably small adult human. The individual known as LB1 stood a little over a metre tall and possessed a brain far smaller than that of modern humans. At first, such anatomy seemed almost impossible for a relatively recent member of Homo. Some researchers initially proposed that the skeleton represented a modern human with a developmental disorder rather than a separate species, but additional discoveries weakened that interpretation. More individuals were identified, and the unusual anatomy was not restricted to one skeleton. The population was eventually classified as Homo floresiensis. Its body proportions, wrist bones, feet, skull, and other features formed a combination difficult to explain as pathology in ordinary Homo sapiens. The evidence increasingly supported the conclusion that Flores had preserved a genuinely distinct human lineage. Its ancestry remains one of the central mysteries. One possibility is that Homo floresiensis descended from Homo erectus populations that reached Southeast Asia, and later became dramatically smaller through island dwarfism. Island dwarfism is well documented in other mammals. Large animals isolated in environments with limited resources can evolve smaller bodies because smaller individuals require less food and reproduce with lower energetic costs. Flores itself once supported dwarf elephants known as stegodonts, showing that strong size changes occurred among other mammals on the island. But Homo floresiensis possesses some anatomical traits that appear unexpectedly primitive, leading some researchers to suggest an ancestry branching from earlier Homo populations, before classic Homo erectus evolved its more familiar body form. Under that interpretation, an even more ancient human dispersal into Asia may have occurred. Scholars still debate which scenario best fits the evidence. Either possibility is remarkable. If Homo floresiensis descends from Homo erectus, then a large-bodied human population underwent an extreme evolutionary transformation in isolation. If it descends from something earlier, then small and anatomically primitive humans managed to travel astonishing distances across Asia, far earlier than previously assumed. The island itself creates another puzzle. Flores has never been connected to mainland Asia by a continuous dry land route during the period relevant to human occupation. Even when Ice Age sea levels fell, reaching the island required crossing stretches of open water. Exactly how early humans accomplished this remains uncertain. Accidental rafting is possible, especially during storms or tsunamis, but maintaining a viable population generally requires more than one individual. Deliberate water crossing cannot be ruled out, although direct evidence for boats from such deep time does not survive. Stone tools from Flores suggest human occupation far earlier than the known Homo floresiensis skeletons. This means some form of human reaches the island hundreds of thousands of years before the population represented at Liangbua disappears. The inhabitants therefore are not temporary castaways. Their lineage survives long enough to become deeply adapted to island conditions. The environment they inhabit is unusual. Dwarf stegodonts provide potential prey. Giant rats share the landscape. Komodo dragons and other large reptiles create dangers entirely different from those faced by Neanderthals in Europe. Homo floresiensis manufactures stone tools despite its small brain size, another warning against treating brain volume as a simple measure of behavioral ability. The known fossils are younger than the earliest occupation evidence, but revised dating has placed the skeletal remains tens of thousands of years earlier than some initial estimates. Their disappearance seems to occur around the broad period when Homo sapiens expands through Southeast Asia, though direct evidence of conflict between the populations has not been found. Climate, volcanic activity, ecological change, competition disease, or demographic vulnerability may all have contributed. No single explanation fully accounts for their extinction. Farther north, another island produced an equally surprising discovery. In Calao Cave on Luzon in the Philippines, researchers recovered a small collection of human bones and teeth belonging to at least several individuals. These remains were eventually assigned to Homo luzonensis. They date to roughly the later Middle or early Late Pleistocene, placing another distinct human population in island Southeast Asia, before the region became dominated by Homo sapiens. Homo luzonensis is known from far less material than Homo floresiensis, which makes interpretation even more difficult. Yet the anatomy is extraordinary. Some teeth are relatively small and show traits comparable to later Homo, while certain finger and toe bones display curved forms resembling much earlier hominins. This combination of seemingly primitive and derived features is exactly the kind of mosaic anatomy that makes classification difficult. The curved bones do not necessarily mean Homo luzonensis lived like a much earlier tree-climbing ancestor. Traits can persist for reasons that are not obvious from a fragmentary skeleton, but their presence shows that human anatomy on isolated islands could follow evolutionary pathways unlike those seen on continents. Luzon poses the same geographic challenge as Flores. It was not simply an extension of mainland Asia exposed during low sea levels. Ancient humans had to cross water to reach it. Archaeological evidence indicates hominin presence on Luzon hundreds of thousands of years before Homo sapiens becomes established there. A butchered rhinoceros associated with stone tools demonstrates that humans or human relatives were exploiting large animals on the island at an unexpectedly early date. Exactly who those early inhabitants were remains unknown. They might represent ancestors of Homo luzonensis. They could belong to another population entirely. Without ancient DNA, which is exceptionally difficult to recover in tropical environments, researchers cannot currently connect these archaeological traces into a clean lineage. That limitation is crucial. Tropical Southeast Asia was likely a major zone of human diversity, yet its climate destroys much of the genetic evidence that would reveal relationships. Heat, humidity, acidic soils, and biological activity accelerate the breakdown of DNA. Bones themselves also decay rapidly. Entire populations may therefore be dramatically underrepresented in the fossil record. The islands become evolutionary laboratories whose experiments are mostly lost. This makes Homo floresiensis and Homo luzonensis particularly important. They prove that unexpected human lineages did survive in the region. Once two such populations are known, the possibility of others becomes much harder to dismiss. During periods of low sea level, the Sunda Shelf connected much of western Indonesia to mainland Southeast Asia, creating a vast landscape of rivers, forests, grasslands, and coastlines now submerged beneath the sea. East of that shelf island, chains formed complicated corridors through which humans and animals sometimes moved. Some ancient populations could remain isolated for tens or hundreds of thousands of years. Others might periodically come into contact. Denisovan-related humans may also have occupied parts of this broader region, judging from the genetic ancestry carried by modern populations farther east. Homo sapiens eventually enters a world that is already populated, fragmented, and evolutionarily diverse. The image of one mysterious lost race therefore becomes less convincing with every island discovery. What emerges instead is a patchwork of small human populations shaped by geography in different ways. Some become smaller, Some retain ancient anatomical traits, some interbreed with newcomers, others vanish almost completely. And because tropical environments erase evidence so efficiently, Homo floresiensis and Homo luzonensis may be less extraordinary than they appear. They may simply be the rare survivors in the archaeological record of a much larger vanished diversity. The next great change in this story comes not from finding another strange skeleton, but from learning how to read the old ones with increasing precision. New dating methods, proteins, genomes, and imaging techniques are beginning to rearrange relationships that once seemed settled, and sometimes a single fragment is enough to redraw the entire human family tree. A human family tree can survive for decades looking reasonably stable. Then one bone arrives and forces several branches to move. This is not because researchers are careless. It is because the surviving record is so thin that each well-dated specimen carries unusual weight. A jaw can change the known geographic range of a population. A tooth can reveal an unexpected relationship. A skull discovered in the wrong place at the wrong time can make an established migration model suddenly look incomplete. The deeper scientists move into the Pleistocene, the clearer it becomes that human evolution is reconstructed from interruptions rather than continuity, and every interruption leaves room for a different history. Dating is one of the reasons interpretations change so dramatically. A fossil without a reliable age can appear to support one evolutionary scenario, then shift into another when improved methods place it tens of thousands of years earlier or later. Archaeologists use several techniques depending on the material and geological setting. Radiocarbon dating is powerful for relatively recent remains, but it reaches only partway into the Pleistocene. Older sites may require uranium series dating, electron spin, resonance, luminescence methods, paleomagnetic evidence, volcanic layers, or combinations of several approaches. Each method has uncertainties, and contamination or disturbed sediments can complicate results. The consequences can be substantial. Homo floresiensis provides a good example. Early interpretations of the Liang Bua sequence suggested that these small-bodied humans survived until surprisingly recent times. Later reassessment of these sediments and chronology pushed the relevant skeletal remains farther back. The population remained astonishingly recent in evolutionary terms, but the change altered how directly its disappearance could be connected with the arrival of Homo sapiens. The broader lesson is important extinction stories can depend heavily on a few layers of sediment and the accuracy with which those layers are understood. Improved chronology has transformed Neanderthal history as well. Researchers once imagined a relatively simple replacement in Europe, with modern humans arriving and Neanderthals disappearing. More precise dating now reveals a complicated regional pattern. Different Neanderthal populations vanish at different times. Homo sapiens appears in some areas earlier than older models expected. Cultural technologies associated with both groups overlap in ways that can be difficult to interpret. What survives in the record suggests a long transition rather than one synchronized continental event. Genetics has been even more disruptive. Before ancient DNA ancestry was reconstructed mainly from anatomy, researchers compared brow ridges, cranial vaults, jaws, teeth, limb bones, and tool industries. Similar anatomy was often assumed to imply close relationship. Genetics revealed that this can be misleading. The Cima de los Huesos fossils in Spain, for example, possess an early combination of Neanderthal-like features. Molecular evidence strengthened their connection with the Neanderthal lineage and helped show how deep that evolutionary history extends. Then Denisova Cave changed the rules further. An apparently insignificant fragment became the first evidence of a major human lineage. Later, another tiny bone revealed the first-generation child of a Neanderthal mother and Denisovan father. Neither discovery required a complete skull. The information capable of altering the family tree was preserved inside fragments that might once have remained unidentified in a collection. This has changed excavation itself. Small bone pieces that earlier generations might have regarded as too fragmentary for detailed identification can now be screened for proteins. One method, often called zooms, examines collagen peptide patterns and can distinguish human fragments from the bones of many animals. Once a human fragment is identified, researchers may attempt ancient DNA analysis. In caves containing tens of thousands of shattered bones, this approach can locate rare human remains that would otherwise be almost impossible to recognize. Sediments are becoming evidence as well. Ancient DNA can survive in cave deposits even where skeletal remains are absent. Researchers have recovered genetic traces of Neanderthals and Denisovans from sediment layers, allowing them to detect population presence and turnover without needing an identifiable human bone. A cave floor can therefore preserve an invisible biological record of people who once occupied it. This is especially powerful because occupation sites are rarely simple. A cave may be used intermittently for tens of thousands of years. Neanderthals may occupy it during one climatic period, Denisovans during another, and later Homo sapiens may arrive. Stone tools can shift styles, animals change sediments, accumulate, and populations return after long gaps. Sediment DNA allows some of these changes to be tracked with a resolution that archaeology alone cannot always provide. Proteins are extending the search into places where DNA survival is poor. Ancient DNA breaks down relatively quickly in warm and humid environments. Proteins can sometimes endure longer. This is why the Siahi mandible from the Tibetan plateau became so important. Molecular analysis of preserved proteins linked it with Denisovan-related humans even without a usable genome. Similar techniques may eventually help classify fossils from East and Southeast Asia that currently sit between established groups. Imaging has also transformed apparently familiar bones. Computed tomography allows researchers to examine structures without damaging rare fossils. Hidden tooth roots, internal ear anatomy, sinus patterns, bone thickness, and the shape of the brain case can all provide evolutionary information. Virtual reconstruction can correct some geological distortion and allow fragments kept in different institutions to be compared digitally. Anatomical traits that were invisible when a fossil was first discovered decades ago can suddenly become significant, yet every new tool introduces new caution. Genetic contamination is a major concern, especially in fossils handled extensively before modern laboratory standards existed. Statistical relationships depend on comparison datasets that remain incomplete. Protein sequences contain less information than whole genomes. Virtual reconstruction involves assumptions about how broken pieces originally fit together. Dating methods provide ranges rather than perfect calendar dates. Scientific precision improves, but uncertainty never disappears entirely. One of the most surprising examples of reassessment involves fossils that have spent years in museum drawers. Excavation is not always required for discovery. Collections contain isolated teeth, unidentified bone fragments and skulls described under classification systems developed decades ago. As new methods become available, old specimens can become new evidence. A fossil labeled archaic Homo may eventually produce a protein signature suggesting Denisovan ancestry. A tooth assigned broadly to Homo erectus may turn out to share internal morphology with a different lineage. The lost human scientists seek may already have been excavated. This possibility is especially important in Asia, where the gap between genetic evidence and recognized fossils remains large. Denisovan ancestry proves that Denisovan-related humans ranged far beyond Siberia, yet secure skeletal identifications remain rare. The mismatch almost guarantees that some currently unclassified or differently classified Asian fossils belong somewhere within that broader lineage. The same could be true in Africa. Deep population structure and possible archaic genetic contributions are increasingly modelled from living genomes, while many Middle Pleistocene fossils remain taxonomically unstable. Connecting the two records will require evidence capable of surviving environments hostile to ancient DNA. Proteomics may help, but many mysteries may remain permanently beyond molecular recovery. There is another complication. A newly identified relationship can simplify one part of the family tree, while making the whole tree more tangled. Proving that two populations mixed means they cannot be represented as completely separate branches. Identifying repeated admixture means lines must reconnect. Discovering that a population itself contains ancestry from an older unknown group adds yet another hidden branch. The result is increasingly unlike the traditional evolutionary diagram. Instead of a trunk dividing into clean species, researchers find networks, reticulations, and populations that separate before exchanging genes again. The fossils have not become less important. They have become more powerful because they can now be read in several different ways at once, as anatomy, chemistry, chronology, proteins, genomes, and environmental context. A small fragment can carry all of these histories simultaneously. And what these methods are revealing is perhaps the most disruptive fact of all. Many of the humans once drawn as separate evolutionary species were not separated by impenetrable biological barriers. They encountered one another, reproduced, and created descendants whose genomes combined histories that taxonomic labels had kept apart. The discovery that Neanderthals and Homo sapiens interbred did more than add a curious detail to human history. It overturned one of the simplest assumptions about ancient human populations, that separate branches of the family tree remained separate once they had diverged. Genetics shows something much messier. Human lineages can remain apart for hundreds of thousands of years, accumulate substantial anatomical and genetic differences, and still meet later with enough biological compatibility to produce descendants. The same is true for Denisovans. Instead of a family tree whose branches never reconnect, the evidence increasingly resembles a network in which old lines separate, cross-merge, and sometimes disappear into one another. When Homo sapiens expands beyond Africa, populations entering Western Eurasia encounter Neanderthals whose ancestors have been living there for an immense span of time. These meetings are not hypothetical. The genetic signatures survive in living people. Most populations with substantial ancestry outside sub-Saharan Africa carry Neanderthal-derived DNA, generally amounting to a small percentage of the genome in any one person, although the combined amount preserved across humanity is much larger. Different individuals carry different fragments, meaning that collectively living humans retain a considerable portion of Neanderthal genetic variation. The mixing does not occur only once. Genetic evidence points toward multiple episodes of contact between modern humans and Neanderthal populations. Some gene flow may even have moved in the opposite direction much earlier from populations related to Homo sapiens into Neanderthals. Human groups repeatedly expand and retreat as climates change, making occasional encounters across long spans of prehistory plausible. The interactions with Denisovans are even harder to reduce to one event. Populations in Oceania carry particularly substantial Denisovan ancestry, while lower levels appear across various Asian populations. The patterns are not identical, suggesting that Homo sapiens encountered different Denisovan-related groups in different places. Some genetic models indicate multiple admixture events involving Denisovan populations that themselves had diverged from one another. This creates a surprising possibility. When modern humans moved through Asia, they may not have encountered a single archaic people called Denisovans. They could have encountered several regional descendants of a much older Denisovan lineage, each adapted to different landscapes and separated by tens of thousands of years of evolution. One population may have occupied colder northern environments, another may have lived on or near the Tibetan plateau, others may have survived farther south in regions where the fossil record is poor. Interbreeding does not mean the populations were identical. Long separation leaves consequences. Genetic studies suggest that much archaic ancestry entering Homo sapiens was gradually removed through natural selection. Some inherited variants may have been harmful when transferred into a different genetic background. Over thousands of generations, these segments became less common or vanished entirely. Other variants survived because they were neutral. A few became valuable. The high-altitude adaptation associated with the Epas-1 region in Tibetan populations is one of the most famous examples of useful archaic inheritance. Denisovan-related ancestry appears to have provided a version of this genetic region that helped people cope with reduced oxygen availability at high elevation. Other archaic-derived variants have been linked to immune responses, skin biology, metabolism, and adaptation to environmental conditions, although the effects of individual variants can be complex and vary between populations. Neanderthal ancestry tells a similar story. Some inherited variants affect immune function, perhaps because Neanderthals had already adapted biologically to pathogens in Eurasian environments. When Homo sapiens arrived, acquiring some of that genetic variation could provide advantages. But other Neanderthal-derived variants have been associated with traits that are not necessarily beneficial in modern environments. Evolution does not preserve genes because they are universally good. Their effects depend on circumstances. This genetic exchange has an important implication for the disappearance of archaic humans. Extinction need not mean complete biological erasure. If a population interbreeds with a larger group before disappearing as a recognizable community, part of its ancestry survives. Over generations, its genes become dispersed across descendants, who increasingly resemble the larger population culturally and anatomically. A small human population can therefore vanish without a single dramatic final event. Imagine an archaic group numbering only a few thousand people scattered across a large landscape. A more numerous population arrives gradually. Some territories are abandoned. Some communities disappear through ordinary demographic decline. Others exchange partners with newcomers. Children have mixed ancestry. Their descendants do the same. After many generations, the original population no longer exists as a distinct social or biological group, even though its genes remain distributed through people who would later be identified as something else. This process is sometimes called assimilation or absorption, though prehistoric reality would have been far more complicated than either word suggests. There may also have been competition violence, avoidance disease, transmission, and unequal access to resources. Different encounters probably had different outcomes. The archaeological record rarely preserves enough detail to reconstruct these interactions at the level of individual communities. One of the most direct glimpses of ancient mixture comes from the individual found at Denisova Cave with a Neanderthal mother and Denisovan father. She demonstrates that contact did not always occur between vaguely related populations separated by generations. At least once, two people from clearly different lineages had a child together. Her father's genome also carried older Neanderthal ancestry, showing that his own population had experienced previous interbreeding. Such evidence makes strict species boundaries difficult to maintain. It does not necessarily mean Neanderthals, Denisovans, and Homo sapiens should all receive the same species name. Closely related species can hybridize, and taxonomic classification considers ancestry and long-term differentiation, as well as reproductive compatibility. But the genetic evidence shows that separation among these groups was incomplete. A lesser-known implication concerns populations that have not yet been identified. If known archaic groups interbred repeatedly, unknown ones could have done the same. A fossil population may disappear from the archaeological record while contributing ancestry to another lineage. Later researchers detect an unexplained genetic signal, but cannot identify its source because the source population has no known genome. This may explain some proposed ghost ancestry. The contributor does not need to survive until very recently. It only needs to encounter another population at some point before disappearing. Its genetic material then travels forward through descendants carrying evidence of a meeting that archaeology may never record. Gene flow can also move through intermediary populations. Homo sapiens does not necessarily need direct contact with every archaic lineage represented somewhere in its genome. A Neanderthal population could first acquire genes from Denisovans or another group. Later, Homo sapiens could interbreed with those Neanderthals and inherit fragments originating from a population it never met directly. Ancient genomes can therefore preserve layers of older encounters nested inside later ones. The human family network becomes increasingly difficult to draw. This also changes how population replacement should be understood. When one form disappears and another becomes dominant in the fossil record, the transition may not represent complete extermination. Biological continuity can persist beneath anatomical and cultural change. Stone tools may shift, body form may change, population size may expand dramatically. Yet fragments of the earlier inhabitants can remain in the descendants of those who replace them. For the mysterious humans, scientists still debate this is perhaps the most important lesson. A population does not need to leave a clear skeleton to matter. It does not need to survive as a separate species until the recent past. It can disappear into another population and become nearly invisible except for molecular traces. But interbreeding alone cannot explain why so many distinct human forms eventually vanish. By the later Pleistocene, the crowded world of several humanities begins to narrow. Neanderthals disappear. Denisovans cease to exist as recognizable populations. Island lineages vanish. Other groups leave only uncertain fossils. The final dominance of Homo sapiens is real, but the reasons behind it are far less simple than a story of one superior species defeating all the others. By the later Pleistocene, the crowded human world begins to empty. For hundreds of thousands of years, different lineages have occupied Europe, Africa, Asia, and isolated islands, sometimes separated by enormous distances and sometimes meeting closely enough to exchange genes. Then, within a relatively short span in evolutionary terms, much of that diversity disappears. Neanderthals vanish as a distinct population. Denisovans cease to exist in any recognizable form. Homo floresiensis disappears from Flores. Homo luzonensis leaves no later trace on Luzon. Other archaic populations survive only as fossils, fragments of DNA or uncertain anatomical signatures. Homo sapiens remains. The temptation is to search for one decisive reason, but the evidence points instead toward a convergence of pressures. Climate is one of the strongest candidates, though it is not a complete explanation. The late Pleistocene is marked by repeated environmental instability. Temperatures shift, ice sheets expand and retreat, rainfall patterns move, grasslands contract in some regions while expanding elsewhere, forests fragment, animal populations migrate or collapse. For small human groups dependent on local hunting territories, freshwater sources and seasonal plants, these changes could be severe. Neanderthals had already survived major climatic oscillations long before Homo sapiens became widespread in Europe, so climate alone cannot explain their disappearance. They were flexible enough to occupy environments ranging from relatively temperate woodland to cold steppe. But repeated instability can weaken populations even without causing immediate extinction. If groups become smaller and more isolated, every additional pressure becomes harder to absorb. Demography may therefore have mattered enormously. Ancient human populations were generally small by modern standards. Neanderthals appear to have lived at low population densities divided into groups that may sometimes have been separated across large areas. Genetic studies suggest relatively small effective population sizes and periods of inbreeding in some communities. This does not mean Neanderthals were uniformly unhealthy or incapable of surviving. It means their demographic margin for error could be narrow. A small population is vulnerable in ways a large one is not. A series of poor hunting seasons can remove essential adults. Disease can spread through a group. Accidents can reduce the number of reproductive individuals. If nearby communities are too distant, there may be few opportunities to replace lost members or maintain genetic diversity. A population can disappear not through one catastrophic event, but through dozens of ordinary failures accumulating over generations. Homo sapiens may have entered some regions with demographic advantages. Larger social networks could connect distant groups, allowing information, raw materials, and mates to circulate across wider territories. Archaeological evidence from later Pleistocene Homo sapiens sometimes shows long-distance movement of shells, pigment, stone, and other materials. Such networks can provide resilience. If one valley experiences scarcity, connections elsewhere can become important. But researchers must be careful not to turn these differences into a simplistic story of smarter modern humans defeating inferior archaic humans. Neanderthals themselves move materials across landscapes, use sophisticated technologies, hunt dangerous animals, care for injured individuals, and adapt repeatedly to environmental change. The difference may have been quantitative rather than absolute. Somewhat larger populations, broader networks, faster innovation in some settings, or greater flexibility under particular ecological conditions. Even small advantages become powerful when accumulated across thousands of years. Competition probably occurs in some places. Homo sapiens and Neanderthals require many of the same resources. They hunt overlapping prey and occupy similar shelters. If modern human populations become more numerous, they can gradually occupy territories previously used by Neanderthals. Competition does not need to involve constant violence. Simply removing prey faster, using landscapes more intensively, or maintaining larger local populations can shift the balance over generations. Direct violence cannot be excluded, but archaeology does not support a continent-wide war of extermination. Injuries exist in ancient skeletons, yet trauma is common among hunter-gatherer populations for many reasons. There is no evidence for a single organized campaign by Homo sapiens against other humans. The reality likely varies from region to region, ranging from avoidance and peaceful contact to competition and occasional conflict. Disease offers another possible pressure. When populations separated for long periods, each may carry pathogens unfamiliar to the other. This process is well documented in much later history, though applying it to the Pleistocene is difficult because direct evidence rarely survives. Homo sapiens moving across large networks might have transported infections between regions, while archaic populations could have exposed newcomers to their own pathogens. Genetic evidence involving immune-related archaic variants suggests disease environments mattered, but the scale of any epidemic contribution remains uncertain. Interbreeding itself may help explain disappearance. If Homo sapiens populations are much larger, repeated mating with small archaic groups can gradually absorb them. The archaic population does not need to be killed or pushed into ecological failure. Its distinctive ancestry simply becomes diluted across a growing mixed population. Over many generations, recognizable Neanderthal or Denisovan communities disappear, even while some of their genes survive. This process may operate alongside competition rather than instead of it. Some communities could be absorbed, others retreat, others fail demographically. Different mechanisms can occur simultaneously across separate landscapes. For Denisovans, the problem is more difficult because their geographic range remains uncertain. They clearly occupy parts of Asia and contribute ancestry to Homo sapiens, but scientists cannot yet identify exactly where their final populations lived or when each disappeared. There may never have been a single Denisovan extinction date. If Denisovan-related groups were widely dispersed, some may have vanished much earlier than others. Island populations face additional risks. Homo floresiensis exists within a restricted geographic range. A lineage confined to one island has no distant refuge if ecological conditions collapse. Volcanic eruptions, changing prey populations, habitat shifts, disease, or the arrival of a new competitor can have disproportionate effects. Flores is volcanically active, and environmental disruptions occur repeatedly in its geological history. Yet linking one event directly to the disappearance of Homo floresiensis remains difficult. The arrival of Homo sapiens in island Southeast Asia is chronologically suggestive, but coincidence is not causation. There is currently no evidence of a battlefield massacre or direct replacement event involving Homo floresiensis. Their disappearance could involve several pressures acting close together. Homo luzonensis is even harder to understand because so little evidence survives. A handful of fossils cannot reveal population size, geographic range, or the exact time of extinction. The lineage may have survived longer than the known remains indicate. Absence from younger deposits is not automatically proof of immediate disappearance. This is one of archaeology's most important limitations. The youngest fossil ever discovered is almost never guaranteed to represent the final living individual of a species. A population can survive for thousands of years without leaving us fossil that scientists have found. Extinction dates are therefore usually brackets, not eyewitness moments. Chance may have played a larger role than humans prefer to imagine. Evolutionary history is full of populations that disappear not because they are inherently inferior, but because circumstances turn against them. A lineage can be well adapted for hundreds of millennia and still encounter a sequence of droughts, demographic losses, competition, or environmental changes from which it never recovers. Homo sapiens may partly survive because of traits that improve resilience, but survival also depends on being in the right places at the right times, maintaining enough connected populations and recovering from crises that eliminate smaller groups. The vanished humans were not failed experiments waiting for modern humanity to replace them. Many survived far longer than recorded civilization has existed. Homo erectus persists for more than 1 million years. Neanderthals endure hundreds of thousands of years of climate change. Denisovan-related populations spread across extraordinary environments. Their eventual disappearance does not erase the success that came before it, and even disappearance is incomplete. When the last recognizable Neanderthal communities fade from Europe, Neanderthal ancestry is already traveling inside Homo sapiens. Denisovan genes are moving east and south through descendants who will populate Asia and Oceania. Other archaic contributions may be carried forward without scientists yet knowing the populations from which they came. The ancient human world narrows to one surviving species, but that survivor is not untouched. Homo sapiens carries fragments of the people it encountered. Their populations disappear from the landscape, yet pieces of them continue moving through time. The vanished humans are not entirely vanished. Their caves may be empty, their tools buried, and their distinctive bodies absent from the living world, but fragments of their biology continue inside billions of people. Every surviving piece of archaic DNA is a small record of an encounter that occurred tens of thousands of years ago. Some fragments arrive through Neanderthals, others through Denisovan-related populations, and still others may come from ancestral groups whose names and physical appearances remain unknown. Modern humanity is therefore not simply the population that survived after all the others disappeared. It is in part a population that absorbed pieces of them. The realization emerged gradually as ancient genomes became available. When researchers compared the Neanderthal genome with those of living humans, they found that many populations outside Africa shared more genetic variants with Neanderthals than could be explained by simple common ancestry alone. The most convincing explanation was interbreeding after some Homo sapiens populations expanded from Africa into Eurasia. Later research refined the picture, revealing multiple episodes of contact and a complicated history of archaic ancestry, being inherited, reshuffled, removed, and occasionally favored by natural selection. No living person carries an intact Neanderthal genome. Instead, different individuals possess different fragments. Recombination breaks ancestral chromosomes into smaller pieces with every generation. Over tens of thousands of years, the original blocks of Neanderthal DNA become scattered through the genomes of descendants. One person carries fragments, another person does not. When these surviving pieces are considered collectively across living populations, they preserve far more Neanderthal genetic variation than any single individual contains. This creates a strange form of survival. Neanderthals disappear as a recognizable population roughly 40,000 years ago, yet portions of their genomes continue reproducing every generation. Their biological legacy travels into societies, environments, and continents they never experienced. Some inherited variants appear to influence the immune system. This makes evolutionary sense. Neanderthals had occupied Eurasia for hundreds of thousands of years before major expansions of Homo sapiens into many of the same environments. During that time, their populations encountered local pathogens and evolved immune responses shaped by those conditions. When newcomers interbred with them, certain inherited variants may have provided useful defenses against diseases already present in Eurasia. But archaic inheritance is not simply a collection of beneficial adaptations. Many Neanderthal-derived genetic segments appear to have been removed from Homo sapiens populations over time. Natural selection can eliminate inherited variants that work poorly within a different genetic background. Some regions of the modern human genome contain unusually little Neanderthal ancestry, suggesting that archaic variants there were disadvantageous or interfered with reproductive success. Other fragments survive without providing any obvious advantage. Genetic drift alone can preserve neutral ancestry. Evolution does not carefully select every inherited sequence for a purpose. Much of what survives does so because population history and chance allow it. Denisovan ancestry tells an equally remarkable story, but its distribution is different. Some populations in Oceania retain substantially more Denisovan-related ancestry than populations elsewhere. Lower levels occur among various groups across Asia. These patterns indicate that Homo sapiens encounters Denisovan-related populations during migrations through the continent and toward island Southeast Asia. The geography of that ancestry provides clues about people whose fossils are largely missing. If Denisovan DNA entered the ancestors of populations moving toward New Guinea and neighboring regions, then Denisovan-related humans must have existed somewhere along relevant migration routes. The precise locations remain uncertain. The genetic map is therefore indirectly revealing an ancient population map that archaeology has not yet reconstructed. One of the clearest examples of useful Denisovan-derived ancestry appears on the Tibetan Plateau. A genetic variant associated with the regulation of responses to low oxygen is strongly connected to Denisovan-related ancestry and became important among populations living at high altitude. Rather than waiting for an entirely new adaptation to arise after reaching the plateau, ancestors of modern Tibetans could benefit from genetic variation, originating in archaic humans who'd already experienced similar environmental pressures. This process is known as adaptive introgression. It is evolution borrowing from another lineage. The concept has profound implications. When populations meet, adaptation does not always begin from zero. Genes tested by natural selection in one lineage can enter another through interbreeding. The recipient population gains genetic variants produced during thousands of generations of earlier evolution. Human adaptation can therefore have a history older than the population currently carrying it. Neanderthal-derived variants have also been investigated in relation to skin characteristics, metabolism, immune responses, sleep patterns, and other biological traits. Researchers must be cautious when interpreting such associations. Modern traits are usually influenced by many genes interacting with environment and lifestyle, and possessing an archaic derived variant does not mean that a living person possesses a simple Neanderthal characteristic. Popular descriptions can exaggerate what individual DNA fragments actually reveal. The deeper significance lies not in any one trait but in the persistence of ancestry. Unknown archaic populations may have left similar legacies. Some genomic studies, particularly those examining African population history, have identified signals consistent with contributions from deeply diverged ancestral groups. Yet scholars still debate whether these patterns require interbreeding with a separate archaic lineage or can be explained by long-standing structure among early Homo sapiens populations. That distinction remains one of the central unresolved problems in the search for lost humans. DNA can reveal that ancestry comes from a deeply separated population without necessarily revealing whether scientists should call that population another species. Imagine two communities isolated for an immense period by changing deserts or forests. Their genes diverge. Their anatomy may also begin to differ. Later climatic conditions reconnect their territories and the populations merge. Hundreds of thousands of years afterward, geneticists detect the ancient division inside their descendants. From the genome alone determining exactly where population difference becomes, species difference can be extremely difficult. What survives in the record therefore suggests not one hidden race waiting to be named, but layers of ancestry produced by populations whose boundaries change through time. Even the genetic legacy visible today represents only part of what once existed. Most archaic DNA has probably disappeared. A small population can contribute ancestry that is later lost entirely through genetic drift. Natural selection can remove other fragments. Some ancient groups may have met Homo sapiens without leaving descendants. Others may never have encountered our direct ancestors at all. The absence of archaic DNA today does not prove the absence of an ancient population. This is why fossils remain essential. Genetics can reveal encounters, but bones reveal bodies. Archaeology can reveal behavior. Geological evidence reconstructs environments. Proteins may connect fossils where DNA fails. No single form of evidence can recover the entire vanished world. Together, however, they reveal something that would have seemed extraordinary only a few generations ago. Living humanity contains genetic material inherited from populations that once appeared separate enough to receive different evolutionary names. The boundary between the survivors and the vanished is therefore not absolute. Every person alive belongs to Homo sapiens, but the ancestry of Homo sapiens is not exclusively its own. The final mystery is no longer simply which lost human population scientists will discover next. It is whether the entire image used to describe human evolution has been wrong in its basic shape. Perhaps there was never a clean tree with isolated branches leading toward one surviving species. Perhaps the more accurate image is a network repeatedly dividing and reconnecting with many of its lines, now invisible but never entirely erased. The traditional human family tree is beginning to look less like a tree at all. For generations, diagrams of human evolution encouraged a particular way of seeing the past. One ancestral form gives rise to another, branches divide, unsuccessful lineages terminate, and eventually Homo sapiens stands alone at the surviving edge. The diagrams were never intended to capture every complexity, but they created an intuitive expectation that human groups could be arranged into separate lines. Ancient DNA, new fossils, improved dating, and the recognition of repeated interbreeding have made that expectation increasingly difficult to maintain. The emerging picture is a network of populations that divide, remain apart, change, encounter one another again, and sometimes merge. This does not mean the human family tree is scientifically useless. Evolutionary lineages genuinely split. Neanderthals, Denisovans, and Homo sapiens have distinct histories that can be traced backward toward shared ancestral populations. Homo floresiensis and Homo luzonensis possess anatomical combinations that justify treating them as distinctive evolutionary groups. Homo erectus represents an extraordinarily long-lived form of humanity spread across enormous areas. Branching remains real. What has changed is the realization that some branches reconnect. Geneticists describe this pattern as reticulation, the formation of evolutionary relationships resembling a network rather than an endlessly dividing tree. Among ancient humans, reticulation occurs because populations that have diverged can later exchange genes. Neanderthal ancestry enters Homo sapiens. Denisovan ancestry enters Homo sapiens. Neanderthals and Denisovans also interbreed with each other. Some genomes contain signals of even older encounters involving populations that have not yet been securely identified. Once these exchanges are included, the search for a single lost race becomes something larger. There may be no one missing population capable of resolving every unexplained fossil and genetic signal. Instead, there were probably numerous groups occupying different places and periods, some sufficiently distinct to deserve species names, and others better understood as regional populations within broader lineages. The terminology will continue to change because new evidence keeps altering the boundaries. Consider what remains unknown about Denisovans. Scientists can reconstruct substantial parts of their genetic history, identify descendants carrying their ancestry and place Denisovan-related humans from Siberia to the Tibetan plateau. Yet there is still no universally accepted complete Denisovan skull. Several Asian fossils could eventually prove relevant, but scholars still debate their relationships. A lineage that unquestionably existed remains physically incomplete. Africa contains a different version of the same mystery. Early Homo sapiens appears to emerge from deeply structured populations distributed across changing landscapes. Some genetic models also suggest ancestry from highly divergent groups, but distinguishing archaic introgression from ancient structure remains difficult. Fossils provide possible candidates without secure molecular connections. A population could therefore be statistically visible in DNA while remaining taxonomically invisible in bone. Southeast Asia adds another layer. Homo floresiensis and Homo luzonensis demonstrate that isolated human lineages survived until unexpectedly late periods. Denisovan ancestry in living populations implies additional archaic encounters somewhere across the broader region. Yet tropical conditions destroy bones and DNA efficiently, while large areas of the Ice Age landscape now lie beneath the sea. Sea level alone has erased an enormous part of the archaeological world. During glacial periods, oceans are lower and coastlines extend far beyond their present positions. Humans frequently favor coasts, rivers, estuaries, and resource-rich lowlands, exactly the landscapes most likely to be submerged when ice sheets melt. Across Southeast Asia, the exposed Sunda Shelf once forms a vast extension of the continent. Other drowned landscapes exist around the world. If ancient populations lived primarily in these regions, much of their archaeological record is now inaccessible beneath sediment and seawater. The missing evidence is therefore not distributed randomly. Some environments preserve human history far better than others. Cold caves can protect DNA for extraordinary periods. Tropical forests destroy it. Dry caves preserve bone better than acidic soils. Coastal settlements disappear beneath rising seas. The human family tree we reconstruct is inevitably biased toward places where evidence survives. This means absence must be interpreted cautiously. A lineage known from one cave may once have occupied hundreds of thousands of square kilometers. A population represented by 3 teeth may have existed for tens of thousands of years. An apparently abrupt disappearance may simply reflect the end of fossil preservation in the sites scientists have discovered. Future technologies will almost certainly reveal more. Ancient protein analysis may classify fossils that cannot yield DNA. Sediment genetics may identify populations without skeletal remains. Improvements in dating will rearrange chronologies. High-resolution imaging will expose anatomical traits hidden inside familiar fossils. Genomic studies of living populations may detect ancestral contributions currently buried beneath later demographic events, and discoveries will continue to come from ordinary excavation. Somewhere in a cave, quarry, river terrace, or museum drawer, a fragment may exist that connects 2 mysteries currently treated as separate. But there is also a limit to what science may ever recover. Most humans who have lived leave no fossil. Most fossils are destroyed before discovery. Most ancient DNA disappears. Languages leave no trace unless recorded. Social identities vanish almost completely. Archaeology can recover a stone tool, but rarely the name of the person who shaped it, the relationships within the group, or the stories people told about their own origins. The lost population scientists debate were not merely anatomical categories. They were communities of individuals. They experienced childhood injury, hunger, cooperation, migration, aging, and death. They learned landscapes in detail. They recognized familiar faces. They probably maintained social traditions that seemed ordinary to them and are now irrecoverable to us. This human dimension is easy to lose when fossils are reduced to specimen numbers. A Denisovan tooth belonged to someone who once moved through a living landscape. The Harbin skull belonged to an individual who saw seasons pass across northeastern Asia. The small bones from Flores belonged to people who navigated an island inhabited by animals unlike those of the continental world. Whatever scientific names eventually attached to these individuals, none of them experienced themselves as transitional forms in someone else's evolutionary diagram. They were simply human in their own worlds. That may be the most important correction produced by recent discoveries. Homo sapiens is not the inevitable destination toward which every earlier population was moving. It is one surviving branch of a much richer evolutionary experiment. Other humans were not unfinished versions of us. They possessed their own long histories, adaptations, and futures, until those futures ended or merged with another population. Scholars still debate how many human species existed during the Pleistocene which fossils belong to, which lineages, and how deeply some ghost populations diverged. Those arguments will continue because the evidence remains incomplete. New discoveries may simplify parts of the story while making others more complicated. The title of the lost human population may therefore never belong to one group. There were many lost humanities. Some have names: Neanderthals, Denisovans, Homo floresiensis, Homo luzonensis. Others survive behind disputed labels such as Homo longi, or within broad categories that future researchers may divide. Still others exist only as statistical signals inside genomes. There may have been populations that left neither fossils nor surviving DNA and are therefore beyond recovery altogether. Yet the fragments that remain are enough to change the central story. Human evolution is not a procession in which one form replaces another until modern humanity arrives. It is a history of isolation and contact extinction, and absorption, adaptation, and chance. Populations separate, become unfamiliar, meet again, and sometimes produce descendants together. Some branches disappear, some survive inside others, and at the end of this immense network stands the only human species still living, carrying within its genome faint traces of encounters with worlds of people who are gone. The landscape is quieter now. The caves that once sheltered different kinds of humans hold sediment, broken stone fragments of bone, and long intervals where nothing recognizable survives. Forests have grown across old pathways. Rivers have shifted away from ancient camps. Coastlines where people once walked lie beneath dark water. The individuals who knew those landscapes are beyond memory, and even the populations to which they belonged often survive only through names invented thousands of generations after their disappearance. Yet their absence is not complete. A fragment of DNA moves silently from parent to child. A tooth remains protected inside a cave. A jaw waits within mineral-rich sediment. Somewhere beneath a museum label written decades ago, an unidentified fossil may preserve the answer to a question scientists have not yet learned how to ask. The deeper human history becomes, the less lonely it appears. There was a time when several humanities shared the earth, each carrying an evolutionary history as real as our own. They crossed mountains and rivers, endured winters and droughts, reached islands, adapted to thin air, and encountered people whose ancestors had taken different paths through deep time. Most of those paths eventually disappear from view. Homo sapiens survives, but survival does not make the past simple. Inside the surviving lineage remain pieces of encounters with Neanderthals, Denisovans, and perhaps populations still without names. Our ancestry carries echoes of a world that no longer exists. Perhaps future discoveries will give some of those ghosts faces. New genomes may reveal where they lived. Forgotten fossils may finally find their proper branches. Species names may change and diagrams will be redrawn again. But there will always be gaps. Beyond the last fossil, beyond the oldest recoverable DNA, there were countless lives that left nothing science can recognize. Their footsteps vanished, their fires cooled, their landscapes changed. And so the mystery ends where deep history so often ends, not with a final answer, but with a faint surviving trace, a fragment carried forward through immense stretches of time, reminding the last human species that it was never the only one.